So I would like to welcome everyone to today's webinar. We are happy that you have connected. And my name is Jaroslav. I'm in charge of marketing activities at Interherence. But we also have our CEO Pierre here with us today. So even the hardest technical questions should find an answer. The topic of the event today is not a specific scientific topic like our previous webinars for cell biology or chemistry of colloids. This time, the common theme is temperature calibration. And you will see examples of that from different fields. We will have three scientific speakers. First, Andrew York will talk about fluorescence biosensors. Then Guillaume Bafou will talk about two different projects, one about measuring refractive indices and also about quantifying thermobleaching, photobleaching. And last but not least, Pascal Lawrence will talk about VAHEAT in the context of light microscopy facility and instrument calibration. We will always have a short Q&A session specific to each talk. And if there are still more questions or there is some open topics to discuss, we can do that afterwards with an open ending. So the questions are best asked through the chat, or you can also raise your hands and ask it after the talk. I want to take a few minutes to introduce our company and the VAHEAT device. So we are a spin-off from the Max Planck Institute in Erlangen, Germany. And you can see our team, which is mostly physicists, engineers, now also some biologists. And we are, by the way, also always looking for new members of the team. We have a couple open positions in Erlangen as well as currently in North America. So feel free to forward to any trainees, for example, that you might have. So what we focus on is making high sensitivity microscopy accessible to everyone through our chip-based technologies. Today we will be talking about chip-based heating and temperature control, but we are also working on chip-based turf microscopy. You can see our chips in the picture below, and our nanofabrication expert Atip in the clean room making them. So that's basically the core of our technologies, always some integrated electronics and optics. And now to the VAHEAT device. So it basically started as a project still at the Max Planck Institute, where Pierre basically found that no existing temperature control solution is good enough for what he needed to do. So he designed VAHEAT. And the beauty of the system is that everything is miniaturized, and both your heating and temperature sensing is integrated into your microscope cover slip. And that results in some unique features of the device. So the temperature control can be really precise. You can achieve high temperatures. You can program any arbitrary temperature profiles. You can go really fast with the heating. Those are all quantitative features, but what people actually appreciate in the end, that the device is really easy to handle, easy to use, mount on a microscope, and it saves a lot of time because there is no calibration time necessary, no equilibration like in these large temperature control systems. To give you a bit more background on these cover slips or these chips, we call them smart substrates, and they have a heated area in the middle made out of a thin layer of indium tin oxide, which heats very evenly the area in the center where the temperature probe is located, which is a platinum circuit that works based on resistive measurement of temperature. So these chips are multi-use disposable. You can recycle them, and you can approach them as normal cover slips in terms of sample preparation. When you have liquid samples, as most biologists would have, you have various options, either from us or we encourage our users to design their own microfluidics and other reservoirs. And that can be all glued on the surface of our smart substrates. So since we have been on the market already for a while, we know that VAHEAT can be useful for researchers from very, very different fields, from biology to chemistry to physics, as you will also see today. And besides such events like today, we also have our resources on VAHEAT. If you are curious and you want to learn more about the device, you can check out our blog post or our recently published white paper. And we spent a lot of time on R&D. The VAHEAT is just a beginning for the company. Our next big project that is now coming to an end, and we will release it, will be this waveguide-based turf system we call QSight. I will not talk further about this, but if you're interested, contact us. We are happy to schedule a call about that. We will certainly keep improving VAHEAT with gas control and also with some cooling option that should also be available this summer. And by user feedback, we are designing new types of smart substrates. For example, one version with larger heated areas. So on the left, you see the version that we currently offer, but soon you will be able to have much larger area where you can place your sample. So that can be useful for people who have much larger microfluidics, or they want to do some spatial biology, they want to place some tissue section on the surface, for example, or they simply want to have really large area cell culture. They want to maybe image more than one sample at the time, which will be definitely possible with our new types of reservoirs. So that's all about VAHEAT and interherence in the beginning. And now I would introduce our first speaker, Andrew York, and give the word to him. So Andrew is a physicist, but currently he is building very useful microscopes for biologists at Calico, which is a company in California, where Andy leads a team. He's well known in the microscopy community for his single objective light sheet microscopes. And today the talk will not be about hardware, but actually about a biological project that Andrew was involved. And so I think I'll stop here and let Andrew talk about the rest.
Thank you so much. Let me share my slides. Okay, so you can, oh, my mistake. Okay, so you can see the slides and hear my voice now, yeah? Very good. All right, thanks so much for the opportunity to share this story. As you said, I represent the microscopy team at Calico, and Calico is a wonderful place to work. It's a Google-funded biotech in the San Francisco Bay Area. And Calico's mission, of course, is to improve human health, and we support that through microscopy technology. But Google-funded, as you might imagine, means we have a substantial amount of intellectual freedom. And one of the ways we like to use that freedom is to try to innovate. We try to invent useful things. And I wanted to tell you about one of my favorite inventions from the past few years from my colleague, Maria Ingaramo, who I believe is also present. So if you have any questions specifically for Maria, she might be able to join in and answer them. And Maria invented a new way to read out fluorescent biosensors that has some really nice advantages compared to existing best practices. All right, so as I'm sure you know, fluorescent biosensors are an incredible technology that reveals otherwise invisible quantities in microscopy images or imaging in general. So on the left here, you can see a mammalian cell, cultured cell, crawling around on a cover slip. But the fluorescent stain you see there, that's a engineered fluorescent protein inserted into the cytoskeleton of that cell that reveals tension, that reveals force as the cell is crawling around. So that's not the kind of thing you'd see in a conventional microscopy image. That's the magic of fluorescent biosensors. The middle panel is my favorite. This is a cultured neuron sitting on glass. But as the neuron is firing, you might expect there to be voltage dynamics. There's a very different, very carefully engineered fluorescent protein in the membrane of that neuron that reveals the voltage as the neuron fires. So if you want to watch thoughts in the mind, this offers a potential path to that dream. And on the right here is kind of the classic fluorescent biosensor, GCAMP. It's a calcium sensor. So in this case, there's a wound delivered to a plant, and part of the response to that wound involves calcium dynamics. And this biosensor reveals the dynamics of calcium concentrations by changing the brightness of the biosensor. So I want to drill down on that. In this case, I'm going to call this a intensity biosensor because the readout is very simply how bright. If the concentration of calcium goes up or down, that'll change how bright is the fluorescent biosensor. And this is often used in neuro work where you're looking at fairly fast dynamics. But for our colleagues at Calico, a lot of our colleagues are looking at much slower processes. They don't want to compare this millisecond to the next millisecond. They want to compare a measurement on Friday to a measurement on Monday, or they want to compare a measurement in January to a measurement in June. And there's a problem when you try to do that with an intensity-based readout for your fluorescent biosensor. And the reason is simple. Brighter or darker could mean that the amount of calcium changed. Brighter or darker could also mean that the laser on my microscope was 10% brighter on Friday than it was on Monday. And maybe you can calculate and calibrate laser intensity, but maybe your sample is expressing 10% more biosensor. The point being, there's a substantial number of confounding factors that are just inherently difficult or really impossible to account for with an intensity biosensor. There's a lot of attempts to try. None of them are really robust. That doesn't mean it can't be done. There is a beautiful technique. Rather than measuring the brightness of a fluorescent biosensor, rather than measuring the intensity, how many photons, you can measure the lifetime of a fluorescent biosensor. And this is a more demanding measurement. It typically requires a more expensive instrument, and current instruments are slower than standard intensity-based measurements. But this is really, really cool because it can measure an absolute quantity, an absolute concentration of calcium. And the mechanism is fairly simple. Instead of counting how many photons did my biosensor produce, you measure the nanosecond interval between when a photon is absorbed by the biosensor and when it's emitted. And you build up statistics for a large ensemble of biosensors, and you can construct this decay curve here. And the cool part is that time interval doesn't depend on how bright is your laser. That time interval does not depend on how many fluorescent biosensors happen to be expressed by your sample. But that quantity does depend on your quantity of interest. So in this case, this is a beautiful example from Joachim Goedert's lab. The nanosecond fluorescence lifetime of his highly engineered fluorescent protein reveals the absolute concentration of calcium. So once you've measured a concentration, that's a measurement that you can compare across days or weeks or months. It's a quantity, not just dynamics. Now, this is beautiful, but I do want to emphasize there are still problems, especially that our colleagues encounter for this type of measurement. And the problem is fairly simple. If anything else in your sample also fluoresces, and for most multicellular organisms, there is substantial autofluorescence, that fluorescence is also in the nanosecond scale. And if you're unlucky, and we usually are, that happens to be the same color as your biosensor. And it becomes fairly difficult to distinguish the fluorescence decay curve from your sensor away from the fluorescence decay curve from your background. And the way this shows up mathematically is it looks like a shift in the lifetime, which looks a lot like a change in the concentration of calcium. So this technique is a beautiful technique for transparent background-free creatures, but it fares, in our hands at least, it fares much more, you get a lot of problems when you start studying anything where there's autofluorescence. They say, what kind of samples have autofluorescence? And the answer is pretty much everything multicellular has substantial autofluorescence. This became painfully clear to us in one of our early collaborations years ago. One of our colleagues here wanted to study aging, and the model organism of choice is the worm, C. elegans. The worm is a beautiful creature for microscopy. It's visually stunning. It's transparent. However, the gut granules of C. elegans are quite fluorescent with no stain whatsoever. They're just inherently fluorescent, autofluorescence. And that autofluorescence is especially strong in the green channel where many fluorescent biosensors operate. And so we would frequently encounter images like this, where I'll tell you that about half of the signal in this image is from autofluorescence, and about half of the signal is from a biosensor. And this is a decent image. This is not like, the worst of the bunch. And the problem you run into here is you can try based on morphology or experience to estimate how much of the signal is background and how much of the signal is sensor. But the fact is, you're not going to pull out accurate quantitative measurements when your signal to background ratio is 1 to 1. So as they say, necessity is the mother of invention. And our colleagues really wanted to make quantitative measurements of small molecule concentrations in living multicellular organisms, living animals. And I invented a way to do that. It's much more work than I'm perhaps making it sound like. But the idea is quite conceptually simple. So conceptually, the idea is, instead of looking at how bright is my fluorophore, or looking at how many nanoseconds elapse between when the fluorophore absorbs and emits a photon, we engineer a special strange fluorophore, an exotic fluorophore that fades spontaneously as you look at it. And we couple that rate of fading to the quantity that we try to measure. And then slightly more concretely, the way we engineer this fading fluorophore is based on photoswitchable fluorescent proteins. So you're familiar with green fluorescent protein, where if you shine cyan light on it, it fluoresces. And there are photoswitchable cousins of green fluorescent protein that have two states. One of them is the standard fluorescent state, much like GFP, but the other state is non-fluorescent. And you can drive the fluorophore in between these two states with light, which is very convenient for microscopy, of course. So one color of light will drive you into the fluorescent state, where you act like a standard fluorescent protein, and another color of light will drive you into the non-fluorescent state, where you don't fluoresce if we shine cyan light on you, for example. Now, for typical photoswitchable fluorescent proteins, the stability of those two states is extreme. So it will stay for hours or even days in the active state, or hours or days in the inactive state. So Maria spent, I want to say, 6 years engineering a special weirdo version of a photoswitchable fluorescent protein that, instead of staying in the active state for hours, it fades out of the active state spontaneously, all by itself, in seconds. That's step one. And it produces a decay curve, which is quite similar to what I showed you with fluorescence lifetime measurements. However, instead of decaying on the nanosecond timescale, which looks a lot like autofluorescent background, this decay is now on the timescale of seconds instead of nanoseconds. And that's critical, because background signals in your sample don't look like that. So all of a sudden, this fundamental ambiguity between signal and background becomes trivially resolvable, because it spontaneously relaxes to the inactive state in seconds. And we couple that rate of relaxation to the quantity that we're trying to measure. So this is a cartoon that I'm showing you here, but here is a measurement. This is a concrete instance of one of Maria's relaxation biosensors. And for this one, the quantity of interest is proton concentration, pH. So this is 5 capillary tubes. And in each capillary tube is the same biosensor, which we call this biosensor pH countdown. It's the first relaxation sensor we made. And each one of these capillary tubes has the same biosensor, but a different pH. And as you can see, we use blue light to drive it into the active state, and then we just watch it fade out of the active state over a matter of seconds. And at basic pH, that fade rate is not too many seconds at all, just a few seconds, and it's faded out. But at acidic pH, 2 pH units later, or 2 pH units lower rather, the fading time drops by almost 2 orders of magnitude. So there's an incredible dynamic range here, and incredible sensitivity to pH with this type of measurement. So already we're seeing some nice advantages compared to lifetime sensing, just in terms of dynamic range. Now, the sample has to hold still for a few seconds, so I'd call that a substantial drawback. But you can measure this on any conventional microscope. In fact, you can estimate these rates by eye, but of course, if you want quantities, you should use a camera and a computer to record data. But arguably the most important feature, as I said, when you vary the background, an intensity sensor gets brighter or darker, just like if you varied the analyte. A lifetime sensor, the lifetime seems to change, even though the analyte might be holding quite stable. But for a relaxation sensor, variation in the background level just looks like an offset for the data. It doesn't look like signal, and that's critical. Whereas when you vary the analyte, all 3 sensors respond in a profound way, and relaxation sensors especially. So the variation in the rate can be enormous, which is really nice. So in a practical sense, that means we become very robust against typical confounding factors in microscopy. So for example, if you just put some ground glass in front of your sample, your resolution of the image is trash. The signal noise ratio of the image becomes far, far worse. But this scattering plate doesn't affect the apparent rate of fading on the seconds timescale. So what it doesn't destroy is the quantitative accuracy of the measurement for inferring pH in this case. Similarly, if you put just an ND filter, an absorber in between your sample and your camera, of course your signal noise ratio is degraded, but that rate of fading is not. So your quantity still comes through in a way that is not systematically distorted. And then arguably most exciting, we are robust against background. So if you just put fluorescent on top of your sample, you get a huge amount of background, but that background doesn't change the fading rate. Fluorescent photo bleaches, but much, much, much more slowly than our biosensor fades spontaneously. So quantitative measurement in an optically nasty sample. And now here in a less quantitative way, but maybe a more evocative way, if you just put meat on top of the capillary tube, you put a millimeter of chicken skin on top of the capillary tubes, the image quality, it turns into garbage. The resolution is trash. The signal noise ratio is trash. You can't even see some of the capillaries especially well, but that fading rate doesn't change, as I said. So we return the exact same decay rates with and without the chicken skin. And this is what I mean by a measurement which is robust against background. I'm telling you it works in capillary tubes underneath chicken skin. What about the worm? Turns out it works great in the worm. So I showed you this image and I said, how do you tell the signal from the sensor is distinct from the signal from the background? And I told you, oh, it's fundamentally impossible with an intensity sensor or a lifetime sensor, or just extremely difficult is maybe an easier claim to defend. But this difficult problem becomes trivial. It blinks. But we drive it into the active state with blue light, and then we watch it fade out of the active state. And since the background neither gets brighter during activation nor fades during relaxation, it becomes mathematically trivial to separate the sensor signal, which I show here in green, from the background signal, which I show here in magenta. Now, I don't mean to imply these two signals are different colors. They're both green. But mathematically, the separability becomes trivial, which is really, really useful for us because now we can take quantitative measurements. And as I said, in this case, the quantity of interest for this biosensor is pH. And we infer the pH, as I said, by looking at the rate of fading of the sensor signal. And at basic pH, it fades very rapidly. At acidic pH, it fades very slowly. And so you can build up this nice quantitative map of pH, despite the fact that the sample is optically terrible for quantitative measurements with any other biosensor we've ever tried. All of a sudden, we're taking robust measurements that we actually trust here. So it works so well in the worm, we wanted to extend this to one of our more difficult collaborations. So some of our colleagues here at Calico work on scientific questions which are relevant for drug development. And of course, the mouse is an extremely relevant creature for scientific inquiry and for drug development. But the mouse optically is a very, very challenging sample. Mice are opaque. Their skin absorbs light, their skin scatters light, and the skin itself is quite autofluorescent. So a signal to background ratio of 1 in the mouse is really excellent. So we did sort of a mock drug trial to really, stretch our technique to its limits. So there's a pH biosensor, and there is a weight loss drug called BAM15, where the mechanism of action of the weight loss drug is believed to be that it interferes with proton transport across the membrane of the mitochondria. Now, Maria's fluorescent biosensor is a fluorescent protein. So we can genetically target that fluorescent protein to the interior of the mitochondria. And if those mitochondria are inside a tumor cell line and the tumor cell line is underneath the skin of the mouse, we can check, can we take quantitative measurements underneath the skin of the mouse in response to a drug? Can we watch how the pH in the mitochondria changes as we inject the drug? And it turns out we can see small changes in the second decimal place of the pH. And these are robust, repeatable measurements. There's actually 2 measurements before administration of the drug and 2 measurements after administration of the drug. But the curves fall on top of each other so well that it kind of looks like one measurement on this plot. And something else that I think is very exciting. This drug, we read a paper about how they established various beliefs about the drug. And one of the things I want to know is, what's the half-life of the drug? What are the pharmacokinetics? And the typical way that I guess pharmacokinetics are measured is you kill a huge number of mice and you do biochemistry on the results. And in this case, the way you do pharmacokinetics is you just wait a half an hour and look at the pH in the mitochondria a half hour later, and you can see it returning to pre-drug baselines. And this mouse went back in the cage afterwards. This is you might call it a non-invasive measurement. The mouse was anesthetized, but we didn't break the skin in order to take this optical measurement. I'm pretty pleased with that. Now, I tell you this measurement is sensitive. I tell you this measurement is powerful. Can we trust it? And this is bringing us to the topic of the day. So this measurement is robust against a wide variety of off-target chemical distractions. So, other things that might be present in the mouse other than pH, are they contributing to the signal? And pretty much everything we tried, the answer seems to be no. Now, this is never exhaustive, but it's quite suggestive. But I'll tell you, the most substantial off-target sensing turns out to be temperature. And this became undeniable for us when we moved our screen from a very warm room to a very cold room, and all of a sudden our experiments took 2 or 3 times as long, which was frustrating but educational and very valuable. And as soon as we understood how critical temperature control was, it turned our results from kind of a mess of non-quantitative gibberish into repeatable, robust science, where we're actually measuring quantities like we wanted instead of just seeing dynamics. And this is really transformative for us. And this isn't so much a product endorsement as it is just the truth. The highest praise I think you can give to a scientific product is it does what it says. And that's what the VAHEAT did. We asked for a temperature and we got it. And since all of our pH sensors are also little thermometers, we would notice if it was wrong, and it's not, which is lovely. And this became especially valuable when we were trying to demonstrate the generality of relaxation sensing. So everything I've shown you so far is pH. There's nothing specific about this technique for pH. We think this is a general mechanism for readout of fluorescent biosensors. So Maria tried to demonstrate that by engineering a calcium sensor. And it turns out that this library that she was screening through, looking for the needle in the haystack, that one hit out of 1,000, we were getting hits on everything we looked at. We were getting hits on things that didn't even contain a calcium binding domain because we thought we'd designed an assay for calcium binding. We ended up designing an assay for thermometers. And it turns out all of our biosensors are thermometers. And when we weren't controlling the temperature, our results were nonsense. And then as soon as we started using the VAHEAT, as soon as we started controlling temperature, we started doing science. So I think that's a good place to end the story. And I just want to note that it was a privilege to work with a really talented team at Calico, led, of course, by Maria, who does not enjoy public speaking as much as I do. But thanks for your attention. And if you got any questions, I'd be happy to answer them.
Thank you, Andrew, for the great talk. So I want to encourage anyone. So we don't have any questions in chat currently. So well, maybe I ask then, what's the direction in the project now? So what are the other analytes that you are focusing on?
This is an excellent question. So we believe we can make an excellent NAD sensor. The truth is, we're quite open to any particular analyte. We like to be driven by the desires of our colleagues and our collaborators. So if there's anybody external who's particularly interested in any particular analyte, we'd be very happy to develop that. And in the meantime, we're focusing a lot of our efforts on the fading time is currently on the order of seconds. And that's fine if your sample holds still for a few seconds. The mouse is kind of wiggly. So one of the directions we'd like to push is we're pretty sure we can push that decay time substantially below seconds and into the milliseconds range. We feel like by laying that kind of foundation for the field, it'll put us in a good position to pivot to any particular analyte that someone wants to study. But a good answer would be NAD. We'd love to make a robust NAD sensor.
Great, thanks.
I have a question on this one related to, the time scales of your biosensors. There is probably a sweet spot where you don't run into the issue of any fluorescence. Fluorescence is fast, but you could have also some phosphorescence background that annoys you. So where do you see the sweet spot of your relaxation time?
So currently it's as fast as we can make it. And the only reason it's not faster is this is where we stopped and published. There is a very interesting dynamic, which it turns out that meat photo switches just a little bit. So mouse skin, chicken skin, muscle, pretty much everything we've looked at photo switches just a little bit and spontaneously relaxes on a time scale that's on the order of 50 milliseconds. So I think the sweet spot for the short term will be as long as whatever time scale we fade at is very distinct from the 50 millisecond time scale. There's no ambiguity and no crosstalk. But I think there'll be this awkward zone as we cross over 50 milliseconds. We'd either want to be substantially slower, which we are, or substantially faster, which I think might take a little while. And we'll see what the, yeah, that's probably a good place to.
Then I have another question, because there's a field of direction where people are actually interested in measuring very local, but in three-dimensional space, the temperature. So I guess if your sensors are not sensitive to other things like calcium concentration, whatever, you make a very specific temperature probe, that could be also exciting. And people using NV sensors, and they do a lot of things to spatially resolve temperature, but it's a lot of work.
We haven't explored that, but I'd be quite interested to explore that, especially if we had a motivated colleague. And you could imagine engineering something which was only responsive to temperature if we can engineer out the pH sensitivity. Or if that turns out to be difficult, you could imagine two different fluorophores, perhaps with two different decay rates. You could look at a double exponential, and by looking at the relationship between the two, you might be able to pick out a point in pH temperature space. But yeah, I'd be real excited to make a good thermometer. This project forced us to become more interested in thermometry than we originally intended.
Great. Thanks a lot, Andy. So I'll introduce the second speaker of today, which is Guillaume Bafou. And Guillaume leads a research group. Oh yeah, he already started screen sharing. So Guillaume leads a research group at Institut Fresnel in Marseille, and he is interested in temperature aspects, different aspects in biology, in physics, mainly in thermoplasmonics, and also on thermophoresis processes. So he was one of the early adopters of VAHEAT, and already did several different projects where VAHEAT helped him. And today he will present probably data from two projects, as you can read in the talk title. So Guillaume, please go ahead.
Thank you for the introduction. Yes, indeed, I have been working in thermoplasmonics for a while now, which is heating gold nanoparticles using light. But I won't talk about that now, because what I would like to focus on is how I have been heating, not using a laser, but using the VAHEAT stage. So yes, I have been using this device for at least 2 applications these last years, and in particular for metrology. And this is what I would like to focus on. So we had 2 applications. One for refractive index measurements of liquids, and the other one for to characterize and quantify the optical properties of materials, the temperature-dependent properties of materials. So the second part is the shortest part. I will start with this characterization of fluorescence molecules. So what we did more precisely was to study the photophysical properties of molecules, fluorescent molecules, and more precisely the temperature dependence of fluorescence, and how photobleaching is temperature dependent. So to do that, we used so this kind of chip. So this is like the naked one, the flat one. We didn't use any reservoir on top. And what we want to investigate is the fluorescence properties, in this case, of nanobeads. So we have fluorescence, not molecules, but nanobeads in the fluid. And we sandwich the fluid between the VAHEAT chip and a 12 millimeter round copper strip. So we put it on top, and then we glue using this picodon twin seal, which is quite a usual, usually used in biology to seal. So we do that, and this way we have like a sandwich with a fluid we want to investigate in between, which is fluorescent because it contains fluorescent molecules or fluorescent nanobeads. And then we place this chip in the VAHEAT stage and in our microscope. And this is a fluorescent microscope. So then we take images of the fluorescent layers we have in this chip. And this is a kind of measurements we have. So we have the fluorescence map, we average all the pixels, and we just look at the fluorescence as a function of the temperature. And this is the kind of measurements we get. So first, on the panel A, on the left, so we just monitor the fluorescence we collect, we measure as a function of time. And with heating off and on, off and on, and so on. So you see that fluorescence is decaying all the time, whether it's off and on, it's decaying. That's because of photo bleaching. And this is what we wanted to quantify. How fast, we wanted to quantify the magnitude of photo bleaching. And as you can see, photo bleaching is temperature dependent. So we expect that we photo bleach our molecules, so we can quantify that. But we can also say how temperature dependent this process is. And the other quantity we wanted to measure is how the fluorescence intensity depends on temperature itself. So I'm not talking about photo bleaching here. Usually when you heat, you decrease fluorescence. And this is also something we could quantify. So it's very weak. The beads are not very sensitive to temperature. So you see the scale here. So the variations are very weak, but still we could quantify that. And we could quantify it using the VAHEAT stage because the benefits are twofold. So compared with an approach with we have our microscope and we vary the temperature of the stage. We have also like a large heating stage. When we did that, we vary the focus when we heat, and this was affecting a lot the fluorescence we measure. So we couldn't get these graphs as precisely as the VAHEAT stage, because fluorescence intensities were varying because of the focus of the microscope. So here the measurements were very precise, and also we are hitting a very small volume because we have a very thin liquid. As a result, when we stop heating, we cool down very, very fast. In a few seconds, we go back to the original background, the ambient temperature in the room. So this was also a benefit. We didn't have to wait like 10 minutes before reaching the steady state temperatures and steady states. So we could have this beautiful shape, slopes on the graphs you see here to precisely quantify all the temperature dependent fluorescence properties. So this was the first application. And the second one was we wanted to measure refractive indices of liquids, of various liquids over a wide temperature range. So to do that, we used the same chip, but this time we used a cover slip on top, same size, but with like a micro crater in the middle. To do that, we just shine a pulse of a nanosecond pulse of light, very intense pulse of light, to make this hole. This is an ablation process. So this hole is like maybe like 100 micrometer in width. So we did that, and so this time the liquid is not fluorescent. This is our liquid of interest. We place this the VAHEAT staged chip, the sandwich structure in the microscope. We have some lights from the top, microscope from below, and this time we don't have any fluorescence camera. We have a wavefront sensor, which is a camera, normal camera. In front of it, you have a diffraction grating, which is a bit special. It's a two-dimensional diffraction grating with a 0 pi checkerboard pattern. 0 pi are phase shifts. So when you do that, I will skip the details, but you can map the wavefront distortion created by your sample. So the sample can be any sample, provided you have like a non-uniform reflective index. So like here, this is a side view. You have an object reflective index n in a liquid of reflective index n0. You have a flat wavefront, and when it crosses your sample, you have a distortion. And this distorted wavefront, delta L, equals the topography times the refractive index difference. So if you measure the waveform distortion, you can measure the refractive index provided you know D, which is the topography of the microcrater, but usually we don't know it. Sorry. So this is the kind of image you get. So usually people using this technique, they are looking at cells, living cells. So this is a cell and this is the kind of image you get. So this is very beautiful and you can learn a lot from these images. So the waveform distortion on the left and the transmission on the right. So you see much more details on the left. But what we did, as I explained, is whether this. So we had a hole within the glass substrate. So when you send the plane wave, you have a distortion. It's the same. So if you measure delta L, you know the refractive index, provided you know this profile of the hole, but we don't know it. So what we do is to do another experiment without the liquid. So we have another distortion, which is different because refractive index is not the same. And what we do now, we have these two distortions in air and with the liquid, which equals these expressions. And then we divide the first image by the second to get rid of this of the topography of the crater we don't know. And this way we have the expression of the liquid, of the liquid refractive index as a function of the two images, two waveform distortions in air and with the liquid. The refractive index of the air, we know it, and the refractive index of the glass, we know it. So we can measure this way the refractive index of the liquid. But this is an image. Okay, we subtract two images. This is an image, but an image that is flat. This is not dependent on the place, the location. So we just average the pixels of the image to have a better signal to noise ratio and a better precision on the measurement. So this was done by my colleague like 3 years ago, but not with a VAHEAT stage. It was done by heating the microscope. We have a microscope where we heat the whole microscope. So it was very slow, but he did it. It was like 6 hours to do that because he had to wait before each point. So we did the same with the VAHEAT stage. And as we can expect, it was much faster. This is a topography of the hole. These are the images we get with a reference sensor in air, in water. So as you can see, the scale are the same. You have a stronger distortion in air. And these are the variations of the profile on the waveform distortion, the radial profile, as a function of the temperature. So from 20 to 70 degrees. And you see they are very small, but we can still measure them. And this is what we could do. So what we did with the VAHEAT stage are the blue crosses, blue dots, the error bars, compared with what was done with the heating chambers we have on the microscope. So first, it's more precise, partly because we use the VAHEAT stage, but also because we improved the post-processing of the results. And also importantly, we could go much further in temperature because of the stage we had that was heating the microscope. We could not heat more than 40 something. So heat, we have no limit on the temperature range. I mean, we can go very far. And also it was very fast. It was like half an hour to get these measurements compared with a full day when we were heating the microscope. So we were very happy with that because we could now take everything we had in the chem lab, the liquid we had in the chem lab, and plot the temperature dependence on the liquid, even mixtures of liquids, for which it's very hard to find databases on the internet, for instance. So this is what we did, for instance, for so water. This is what I presented. Glucose, 10% in water, ethanol, and this polymer. A colleague of ours needed to know the refractive index as a function of temperature for this polymer, and we could measure it like very fast, and it was very precise. And also, so this is the next step. We started, but we are working on that now. Within your sandwich, you can put any liquid, but you can also put any mixture and any fraction of the mixture. And also we can vary the wavelengths of the illumination. So we can build a map of N as a function of T, temperature, concentration, and wavelength. So for instance, this is what we have done here for glucose concentration. So we have like a map of N values as a function of T and C. And again, because this is very fast to plot one curve like that, it's not prohibitive to build this kind of map. You can build this kind of map within a day, for instance. Yeah, so this is the benefit we see of this device, and this is what we are doing now, to have complex fluids and vary many parameters and study, measure N as a function of all of these parameters. So everything I've presented today was mostly the work of my previous PhD student, Salman Shakib. And the take-home messages are, so we used the VAHEAT stage as a metrology tool in optics to measure optical properties that are dependent on the temperature, including fluorescence. But in this case, you need to couple the VAHEAT device with a fluorescence microscope. And so, yeah, I inversed the two parts of my talk, and also to measure reflective indices. And in this case, you need to couple the VAHEAT device with a wavefront sensor. Thank you for your attention.
Thanks a lot for your talk. So we have time for one or two quick questions. Okay, so you also used VAHEAT in one project where you grew some thermophilic microorganisms. Is this something that you think about working on in the future, or that was just one isolated project?
Yeah, so I didn't talk about that because it's not really metrology, but yes, we are using also the VAHEAT stage for biology to heat thermophilic bacteria, for instance. So yes, so this is working well. We are fighting a bit with the environment. For some bacteria, they need oxygen. So we built, so in this case, we use this reservoir, and we built like a triangle plate, very small triangle plate that we put at the bottom of the reservoir to have a thin layer where the bacteria are, but also enabling oxygen to flow and to be exchanged easily with the bacteria. And for another project, our bacteria do not like oxygen. So in this case, so I'm very interested in what you could do, like the microfluidic chips where gas can be controlled. I'm very interested in that. It could help us investigating this kind of bacteria. Yeah.
That's true, that there's a lot of anaerobic archaea, for example, that people are very interested in culturing and in imaging.
I have a short question to your last part of the talk. First, thanks for your talk. What kind of why is it interesting to measure the temperature dependence, or as you're saying, like concentration dependence of the refractive index? What is the kind of is there an application?
Okay, so I didn't talk about that, but I'm using this reference sensor also to measure temperature gradients, because when I hit with my laser my field of view, the center of my field of view of the microscope, I create a temperature gradient which distorts the plane wave. And by measuring this waveform distortion, I can retrieve what the temperature is, provided I know the dN over dt. I know how the reflective index is changing as a function of temperature. So I know how to use this technique in water, because we know the dN over dt of water, but for some applications, I need different fluids, mixtures of fluids, and for these fluids, there is no database on dN over dt. So I need to measure them.
Okay, thanks a lot. So
Thanks.
let's move to the last talk of today, which will be given by Pascal Lorenz. Pascal is a facility manager in the microscopy core at the University of Basel in the biomedical department. And so he will give us a slightly different perspective on using VAHEAT in a microscopy facility. So he received his unit relatively recently, but he was doing a lot of calibration measurements on his many microscopes. So stage is yours, Pascal. Thanks.
Thanks a lot. Can you hear me? Yeah, okay, perfect. Yeah, so thanks for the introduction and also welcome from my side. As Jaroslav already said, my name is Pascal Lorenz. I'm heading a microscopy facility at the Department of Biomedicine at the University of Basel. If you want to follow us, here are the tags for Twitter and LinkedIn. During my short talk today, I will mainly talk about how we used VAHEAT as a measuring device. But before we look at the results, I would quickly like to show you who we are and also how we normally perform live imaging experiments where we actually use temperature or controlled environments. And then finally, we will end with a short summary and an outlook. So the Department of Biomedicine is a joint venture of the University of Basel, the University Hospital Basel, and also the University Child's Hospital. And this actually unites the biomedical research of the Faculty of Medicine from the University of Basel. And the goal here is to create a fruitful environment for translational research where clinicians and non-clinicians work closely together. Our department is spread over 5 different locations all around Basel, and the microscopy facility is located in 2 different locations. So we have 2 different sites. There is one at Hebelstraße and there is another one at Mattenstraße here. Currently, we are planning a new building, which then should combine all the different research groups in one building in 2029. That's at least the plan. And here you can see a visualization from the architects. The facility actually offers a large variety of equipment or high-end systems ranging from confocals to slide scanners, high content screening, but also laser microdissection. The department itself has around 850 employees divided into 70 research groups roughly. As I said, spread over 5 locations, and they perform research in 4 different research clusters. And this creates around 300 users for the microscopy facility with around 40,000 booking hours a year. So why VAHEAT? As you know, this device, we heard a lot already about it. This is a temperature, or it's a device that you can use for dynamic temperature control, and it has a great stability actually. And I got to know this device last year at a workshop at the ELMI meeting. And I was pretty much impressed by the capabilities that this device offers, and also about the preciseness, and also the possibility that you can actually not only control the temperature, but also measure the temperature. So because there is always this feedback loop, of course, at the level of the sample, because so far this was not possible for us to really measure the temperature at the level of the sample. On this slide, you can see here how as a standard live imaging setup looks in our facility. So we also have these incubation boxes around the microscope. So we actually then have this heater on top of the shelf, and then we heat up the whole microscope. The advantage here compared to a stage top incubator is that the whole system is at 37 degrees. And that also allows you to compensate a little bit for temperature drifts within the room if your ventilation goes on, or if your air condition kicks in. Also overnight, sometimes they are reduced during off hours, and then we might have slightly temperature changes in the room. So this actually keeps the whole system stable. Then to do for the humidity and for the CO2 concentration, we have these gas mixes. This is the red unit here. The gas, so we can set the desired CO2 concentration, and then the gas is mixed together with air and is then blown through a water column here, which also sits inside the temperature box. So it's also heated up to 37 degrees. It has even an additional heating stick, so you can even heat it up higher, so you get more humidity into your gas. And then it's actually from there, it's then blown into the stage top incubator that sits on the stage and is then a closed kind of an environment or box, which then contains the humidity and the CO2 concentration. What you can also see here is the temperature sensor, which is, as you see, very close to the sample, but still it's not inside this stage top incubator. And in fact, it's quite far away from the sample itself. And this was, I mean, so far this is how we measure the temperature. But of course, it's not, let's say, super precise. And so using the VAHEAT device, actually, when I saw this first at the ELMI meeting, I had the idea to kind of misuse it as a measuring device because it's so precise and it really allows to measure the temperature on the level of the sample. And when I got contacted by Interherence, if I would be interested, and I told them, yeah, I don't have a clear application where we would need temperature changes or quick temperature changes, but I would like to use it as a measuring device. They kindly offered to provide a demo system. And so we used this to do some measurements. And we came up with a few questions. So first of all, we wanted to see actually how long does it take until this box is really on a stable temperature. We then also wanted to know, do we actually reach the temperature that we set? So it's to kind of control the temperature sensor that is inside the box. And then, of course, we wanted to know if this temperature is stable over a longer time period, because normally we do these experiments overnight or even longer. So we need to know if this is really stable. And then we were also interested in how illumination would change the temperature of the sample. So if you then actually go with light onto the sample, does this change the temperature? Then, of course, we looked at different systems like wide field, but also confocal, or like point scanning confocals and also spinning disk confocals. So as I said, first of all, we looked at how long does it take actually when you switch on these heating units to get the whole box onto a stable temperature. And we could see that in some cases, the plateau is reached faster than on other devices, but roughly it takes around 60 minutes to get a stable temperature. So we measured several of our microscopes, and these are just two plots that I show here. We then wanted to know, of course, if the desired temperature is actually reached. And here we found that in some cases, we actually get slightly higher temperatures. Instead of 37, it's around 0.3 higher. And then in other cases, we did not even reach the 37 degrees. So we were quite far away, almost a degree away from the set point. And then, of course, we could use this information to slightly adapt our set points so that we then finally reach the 37 degrees. We then looked at longer time periods. So in that case, we performed a measurement over 28 hours. And we saw also on the other systems that once the temperature is reached, it stays rather stable. So that was actually fine. But what we also found on one of the systems is this pattern. So this was quite surprising. And if you look at it a bit more in a different scale, we saw that there is a regular temperature cycle of around 60 minutes where the temperature drops to almost 35 degrees and then it goes back up to 37 degrees. So and yeah, we thought that this is probably should not be the normal case, because in all other cases it was stable as you see it here. And so we definitely had to get this unit checked. So then finally we looked at illumination, or if light actually has an influence on the temperature of the sample. We used water in a reservoir. I mean, this is was used as an easy to use sample, but it's of course not a real sample. And we know that a real sample might have a different light absorption, so could have a different behavior. But we just used water as a easy to go, liquid. We illuminated the whole thing with in a live view with 30 seconds with full power light that the system or whatever system we measured delivered. And we saw that in wide field cases that the temperature can go slightly up, so it increased like not a degree but almost a degree, while on point scanning confocal but also on spin disk confocals the temperature didn't increase at all. So this was also a little bit surprising because I was expecting the opposite, to be honest. But this is probably due to the fast scanning and also that we only illuminate a small point, while on a wide field system you illuminate a larger area at a time, and if you then have it live for like 30 seconds, that's still quite a long time. So that's at least what we assumed. What we also found is that you have to be careful actually that you do not measure directly on the sensor. We saw this scheme now several times today. There is this little temperature sensor, and if it happened that you illuminate this sensor directly, then the temperature can increase quite fast to 50 degrees or even higher, because the sensor seems to absorb the light pretty well, and then the temperature increases. So make sure if you use fluorescence, I mean, then it can happen that you don't see the sensor. So it's recommended to use transmitted light techniques to find the sensor and then move it outside of your field of view. So this is actually what happened at the beginning. So we were accidentally measuring on the sensor, and then we had quite high temperatures, and then we were a little bit surprised. So this is, I think, is an important point. Yeah, so as a short summary, what we found is that it takes around 60 minutes for our systems to get to reach a stable temperature, and that the temperature that we set is not always what we get. So we have to actually compensate there or be careful. And then in terms of illumination, we found that wide field illumination can increase the temperature of the sample, or at least the sample we used there. And also at that point, scanning and also spin disk confocals didn't change the temperature at all. And as an outlook, we would probably like to check different objectives. So we were wondering if different numerical apertures have an influence because they focus the light differently onto the sample. Then it would also be worth to check different wavelengths. So if they have a different, or if we see a difference in heating. And then finally, it would also be, of course, ideal then to use a real sample that has a certain light absorption, and this might again have an influence on the temperature. With this, I'm already at the end, and I thank you for your attention. Happy to take your questions if you have any.
Thanks a lot for this very interesting and detailed data. So Misha Mitkovsky in the chat has a question. Can you describe a bit more what objective was used during the wide field confocal temperature measurements, oil immersion, etc.? Did you measure?
Yeah, we used low magnification in that case. We used 10x only with air, so no immersion actually, because this was also something that people did actually, checking for when you have a stage top incubator and you heat only your sample, and if you then connect the oil immersion objective, you have a kind of a heat sink. And so this is why sometimes people use heating collars on objectives to compensate for this. But in our case, we didn't look at this because we have this whole box around the microscope, and then this keeps the whole setup at 37 degrees. So that's why we didn't look into this. Was there anything else? The confocal oil immersion? No, we didn't measure the laser power. We just boosted it to on confocals. I think I used 50% laser power of what the system delivers. But I agree, it would be interesting, of course, to measure the light dose actually that you put on the sample, also on the wide field, and then you can probably calculate on the area that you illuminate.
So maybe would you say that routine temperature checks could be another thing that the facilities should do, like they measure the laser power from time to time. So
yeah, I guess it's a good idea to check your temperature devices, because as we saw, in some cases we do not reach the 37 degrees. In other cases you might have like 38 or whatever degrees. This didn't happen for us, but you can imagine that this can happen. Then if you work like this with your cells on 38, that's probably not what you want. But also if the temperature is too low, then you might have some delays in dynamics when you have colder temperature. So yeah, of course, I mean, what we did sometimes is we put a probe inside the stage top incubator and we measured. But again, this is not on the level of the samples. So I think this device is pretty nice to, also as a measuring device. It's just pretty useful, yeah, and to do a kind of a calibration for your heating device.
Thanks, Pascal. And so if there are no more questions, I'll just remind everyone that you can reach out to us if you're interested in VAHEAT, or if you want to discuss more, and also reach us if you're interested in the turf system QSide, and we will be releasing it in summer. So follow us on social media or check our website from time to time. There will be news. So yeah, we will be very happy if you get back to us after this event, and we are happy to discuss temperature or anything else. And I would like to thank again all the speakers. That was a very interesting discussion today, and I think very relevant topics on reproducibility or getting really quantitative information from your imaging experiments. So I think this needs to be discussed more. People often ignore this. So I think we will get back to this in future again. So thanks everyone.
Yeah. Thanks also from my side for the talks. Was great seeing your presentations, and I think I got a few ideas of what could do in future. That's always really cool. So thank you, and have a nice day or evening, depending on where you are.
Bye.